Good evening. I know almost all of you now, so I'm Caroline Gelb, and I, along with my colleagues, are delighted to welcome you to this month's Community Talk series hosted by Our Future is Science. This is part of the Mentorship and Scholars Program of Our Future is Science, and we are happy to welcome the public as Zoom allows us to democratize these programs. OFIS is a joint program with the Aspen Institute. And as an organization, we are committed to sparking curiosity, passion, and confidence about STEM in young people to push science forward in the 21st century and solve society's most pressing issues. We are generously supported by Amgen Foundation, the Samuel I Foundation, Burroughs Wellcome Fund, 3M, and the Carson Family Charitable Trust. Without these funders, none of this would be possible. So the Community Talk series is a platform to expose individuals to diverse careers, as well as information, insights, and perspectives on the intersection of STEAM disciplines and social justice issues. The title of tonight's talk is Unpacking the Science of Infectious Disease and Public Health Disparities. And we are delighted to feature two stellar scientists at the start of their careers studying infectious diseases and epidemiology. We have Dr. Sydney Stanley and Dr. Jelonia Rump. I will turn it over to Hassan, who will introduce our first speaker.
Great. Thank you, Caroline. So I'm extending a warm welcome to everybody in Zoom world today. Good evening. So I'm thrilled to introduce tonight's first speaker, Dr. Sydney Stanley. Sydney is a postdoctoral research fellow in the Division of Infectious Diseases at Boston Children's Hospital. She's also a member of the Computational Health Informatics Program at Boston Children's Hospital. Dr. Stanley is interested in mitigating global health disparities caused by infectious diseases. She does so by leveraging bacterial population genetics to improve the efficacy of vaccines, antibiotics, and diagnostics. Currently, Dr. Stanley is employing comparative genomic techniques to understand how some serotypes of Streptococcus pneumonia evade vaccine control. Dr. Stanley earned her Bachelor's of Science from Duke University, where she majored in biology and global health. She completed her Ph.D. in the Biological Sciences in Public Health program at the Harvard School of Public Health. Her dissertation work was centered on Mycobacterium tuberculosis, evolutionary genetics. Welcome, Dr. Sydney Stanley. Thank you for your patience, everybody.
Thank you for that introduction. I'm going to share my slides now. Hopefully that introduction will make a little bit more sense after this presentation. I'm just getting situated so I can see if I can see. Or do you guys see the slides okay?
Yep.
Okay, yeah, sorry, I can't see your video in this format, so. Okay, so I'm going to go ahead and get started then. This was mostly covered in the introduction, but I was born and raised in Greensboro, North Carolina, and so I eventually went to do my undergraduate work or study at Duke, and as mentioned, I majored in biology and global health, and then afterwards I went to the Harvard School of Public Health for my PhD, and I studied biological sciences and public health. And as mentioned, I'm currently a postdoctoral research fellow at Boston Children's Hospital. And so I thought I would include a schematic of like the different microorganisms that I spent time researching over the course of my career. So in undergrad, I did a lot of work on viruses, mostly on cowpox and Zika virus during that outbreak. You guys may be too young to remember that, but that was kind of like the COVID of the day when I was in college. And then my PhD work centered on Mycobacterium tuberculosis, but I also spent some time researching SARS-CoV-2 during the pandemic. And then now my research focuses on Streptococcus pneumoniae. So if you caught the title of my presentation, we're discussing infectious diseases and global health. So I thought we'd start with the basics. And so we can have like an understanding that'll make this connection make a lot more sense. So what are infectious diseases? So they're illnesses that are caused by microorganisms. So they're illnesses that make humans sick, and the causative agent are also alive. So this includes bacteria, viruses, fungi, and parasites. And so a key characteristic of infectious diseases is that they're transmissible. So they can spread from person to person, like through blood or through airborne, like if you cough, or through vectors such as mosquitoes or ticks. And infectious diseases are largely preventable with vaccines, and they're largely treatable with antimicrobials or drugs. And these characteristics are important because they shape the outsized role infectious diseases play in global health disparities. But we'll talk about that a little bit more later. And so that brings us to global health. What is global health? So global health is the field of research and practice that is concerned with improving health and health equity for all people worldwide. And this is kind of distinct from public health, which is concerned more with defined communities. So with public health, you may think of like you have like public health officers that act on the city level, the county level, maybe the state level. Global health, again, we're thinking about global communities. So we're thinking on the scale of nations or continents and regions. Also, at the heart of global health is the concern with equity. That is like the motivator, that's the mission of global health, is to increase health equity across the world. And again, that's not necessarily the mission of public health, even though that can be an outcome. So infectious diseases are a major driver of global health disparities. And what I mean by that is that the burden of infectious diseases are not distributed equally across the world. So I'm showing you a map here, and it shows the incidence of tuberculosis. So incidence is like the number of new cases. And tuberculosis is a terrible lung disease. It's caused by this bacteria. It results in 10 million cases of disease each year, and this leads to 1.5 million deaths. And so I don't know if you remember, but at the height, COVID was killing 1.5 million people a year. So there's like a COVID pandemic occurring every year for the past centuries that people kind of don't care about. And the reason why we mostly here in the United States don't care about it is we don't see it in the United States. So in this map, you can see the regions that are in blue. Those are the regions that have the largest burdens of tuberculosis disease. And the regions in white and black are mostly developed countries like the United States, Australia, Western Europe, see a lot less of these cases. And I'm just showing one disease here. This is tuberculosis, but this is pretty much true for all major infectious diseases, where you see the largest burden of disease in the poorest countries. And this trend is like so entrenched in the way our society works, to the point where you can actually track a country's stage of economic development based on the diseases that people die from. So countries that are like poor or not as economically developed will experience a lot of deaths from infectious diseases. And so those are countries that are in the beginning stages of their epidemiological transition. But as countries develop and they become more economically advanced, they experience deaths from chronic diseases or diseases of old age, like heart disease and cancers. And so both sets of diseases, like the main killers in different countries, is a reflection of the environment of those corresponding countries. And so it's kind of a two-way street. So if you don't have access to clean water, you're going to be exposed to infections that are spread through dirty water. So poverty is leading to infectious disease exposure. But if you're sick all the time with like debilitating disorders, how do you get out of poverty? And so I'm showing that here with cholera. It's spread through the water. It causes a lot of morbidity and mortality, and it worsens poverty in a lot of different regions. It's same idea with malaria. It causes a lot of disease in children. So if you're a child sick with malaria, there's a good chance that you might not live long enough to get cancer. you're not nourished enough to get diabetes. So the association between the economic development of a country and disease makes a lot of sense. And so that is mainly how we think about global health disparities, is the increased burden of infectious diseases in the poorest parts of the world. And so this brings me to how I got interested in studying infectious diseases. So when I was in elementary school, I think it was, maybe middle school, I read a letter from a Birmingham jail by Martin Luther King Jr. And one of like the most famous quotes from that letter is, "Injustice anywhere is a threat to justice everywhere." And of course, he was talking about civil rights, but this is also very apt regarding infectious diseases. So, like, quite literally, infections are transmissible. They don't respect national borders. So, like we saw with COVID, an outbreak in one part of the world can affect health across the globe. But this also applies to the unfairness of infectious disease. I think it's just fundamentally wrong that millions of people are dying from diseases that are largely preventable with like clean water or soap. People are dying from diseases that we in the United States have vaccines and drugs for. And these are diseases we mostly hear, people here probably haven't even heard of. And so I just have always found that fundamentally wrong. I was interested in infectious diseases um, for a while. I just thought microbes are cool. I thought it was cool looking under a microscope and seeing these weird bugs. And it was just profound to me that these organisms that you can't even see are able to shape so much of our lived experiences. But it wasn't until like I read this quote and then later on studied global health in college where I understood like the impact infectious diseases were driving, and not only in health disparities, but also economic disparities across the world. So I would say that's like the main motivator for why I got involved in the research that I do now. So lots of different factors intersect in this interplay between infectious diseases and global health. So we already talked about poverty and access to clean water. But in addition, the warming climate expands regions in which mosquito-borne infections can spread. So also thinking back to that map, a lot of like tropical climates, warmer climates, you see a lot more disease because there are a lot more mosquitoes. We see limited education. It impairs the ability of people to make sound health decisions. So regions in which either women or children don't have access to education, you're going to see a larger burden of infectious diseases. If you're malnourished, you're going to be more prone to infection. If you have inadequate health care infrastructure, you're going to miss cases of diseases that are easily treatable and easily preventable. Also, with overcrowding, like in slums, if you have a lot of people and small spaces, then it's easier for airborne diseases to transmit. And then also we see with war, war is known to destabilize a lot of these different sectors of society. And so war is also a huge driver of infectious disease spread. We see a lot like with destabilization in the Middle East. Whenever there's conflict, there's more polio and there's more cholera. It happens every time. And then also we see in the war in Ukraine, there's an outbreak of tuberculosis, multi-drug resistant tuberculosis actually. So the aspect the aspect I actually focus on is the lack of access to vaccines and antimicrobials. But more specifically, I think about how vaccines and antimicrobials are ineffective. And I try to find ways in which we can make them better at doing their jobs. So in order to do that, I employ a bacterial population genetics approach to inform ways in which we can make antibiotics and vaccines better. So this kind of follows, this research can be kind of like summed up into 3 steps. So the first thing I do is sequence pathogen genomes. So like I said, pathogens are microorganisms, they're living things, they're they're organisms, and so they have their own genomes. And so what I do is sequence the genomes of pathogens that are infecting people. And so in this case study or example, we're going to focus on tuberculosis. It's a bacteria. So pathogens are genetically diverse. And we find that different variants infect different regions of the world. However, we employ a one-size-fits-all approach to how we treat infectious diseases. So everyone in the world is getting the same treatment right now for tuberculosis. Even though we know that there's diversity in the bacterial populations. So what I try to do is I employ a comparative genetics approach where I try to identify mutations that distinguish pathogens from different regions of the world. So in this schematic here, I'm showing like we've defined a set of mutations that are common in the microbacteria that affect people in Asia. And like by doing so, we also are finding mutations that are distinct elsewhere in the world. So in this example, these mutations, for example, are found only in the mycobacteria that affect people in Africa. And then after I find those mutations that are common within different regions, I employ experimental validation to see if these mutations actually affect the efficacy of antibiotics and vaccines. Because if they do, then we can tailor our treatment to optimize outcomes for each population. So for this example, and just like in this schematic, based on the mutations found in this microbacterium that's common in Asia, this antibiotic should work, but this bacteria is going to be resistant to this antibiotic. So in this region of the world, we should only use this antibiotic. Conversely, let's say in Africa, only this antibiotic should be used. But that is not the paradigm of how we treat infections right now. And so that was like a hypothetical example, but this was the heart of my PhD research, where I focused on Mycobacterium tuberculosis, or NTB strains from Vietnam. And then I found mutations that distinguish strains from Vietnam. So we can just divide them into 3 different groups based on their mutation profile. And what we found actually was that strains from this group, we can call them L1 strains, were more sensitive to these 2 drugs. They're called bedaquiline and clofazimine, but they're really resistant to protomidid. But like I said, everyone in the world right now are getting these exact same sets of drugs for TB. And this matters because Vietnam is a high TB burden country. So there's a lot of TB cases in Vietnam. But we aren't optimizing what we know to treat TB better in Vietnam. And what's really interesting is that Thailand is very close to Vietnam. We talked about how bacteria spread, but these strains that are super susceptible to these drugs are only found in Vietnam, actually. They're not found in Thailand. So right now, Vietnam and Thailand are being treated with the same antibiotic regimen, even though we now know that we could tailor treatment to optimize treatment outcomes for tuberculosis in these different regions. And Thailand's also a high burden TB country. So that's kind of like the theme of my work. I try to find opportunities for tailored region-specific treatments so we can improve health outcomes for the people who need it the most. And so that's like the area in which I kind of intersect, but global health, like by definition, is very interdisciplinary. So we need people acting in all different sectors. So something to think about is like where and how can you act, or at what nodes could you act to mitigate global health disparities for diseases that you may care about. That's what I have for now.
Thank you so much, Dr. Stanley. All right, fantastic. That was a wonderful look into how infectious disease and public health disparities are affecting individuals, not just here domestically, but globally. And it is really nice to see. We're going to transition very briefly over to Dr. Jelonia Rump, who's going to be joining us as well. So Dr. Jelonia Rump has joined OFIS as a mentor in our first cohort in 2021. She also continued in the program as an ambassador last year, so it's great to have her back. She also graduated from Harry Medical College with a Ph.D. in biomedical science in 2023. After defending her Ph.D., she worked as an environmental epidemiology postdoc, a fellow at Emory University. In July 2023, she was commissioned into the U.S. Public Health Service as a lieutenant. Lieutenant Rump now works as an Epidemic Intelligence Service Officer, the National Center for Environmental Health at the Centers for Disease Control. Welcome, Dr. Rump. The floor is yours.
Hi, thank you. Let me just share my screen. One second.
If anyone else has questions, you guys can add them in the chat and we will ask you to unmute a little bit later at the Q&A session, everybody.
Oops. Okay, can you all see my screen?
Yes.
Okay, Okay, so as Hassan said, my name is Jelonia Rump, but I usually go by Loni, and today I'll be giving a talk about how I merged public health and basic science in my early career, and just kind of giving some insights on ways that hopefully you all will be able to do that as well. And this is just a disclaimer that the thoughts in this PowerPoint are my own and not representative of any of the organizations that I am associated with or have been associated with. Hold on. So as I said, I typically go by Loni. I'm originally from Orlando, Florida, but since then I've lived in Miami, Nashville, and now I'm actually in Atlanta. I did earn a PhD in biomedical science from Meharry Medical College. I currently work as an epidemic intelligence service officer at the CDC. And just a fun fact about myself is that I make and sell skincare products for sensitive skin. So first, I want to just go over my educational journey. And that, of course, well, I guess my educational journey that really started leading to my early career started in high school. And this is because my freshman year, I actually joined a medical magnet program at Apopka High School. And I was able to take like health science classes. I was able to take AP biology. I was able to participate in like clinicals and things. I think I went to like a nursing home and a fire department or like a fire station to volunteer during that time. So in high school is when I was really able to develop or figure out that I was interested in health science. And at that time, you know, my plan was to become a nurse. That was kind of the main job that I knew you could do in the health sciences. But after graduating from high school, I went ahead to Florida Memorial University, and at Florida Memorial University, oops, hold on, my computer froze. Oops. Okay. So at Florida Memorial University, I did major in biology. And basically each summer that I was in school, I participated in like a summer enrichment program or like a summer research program. And I'll go over some of those in a later slide. But during this time, basically because I did those summer programs, I was introduced to public health. So going into college, I didn't actually know what public health was, or, that it was really something that I could even major in. So upon finishing my undergrad degree and then learning about public health and then doing research for 3 years, I decided that I would pursue a career as a researcher. And that led me to attend Meharry Medical College. So, excuse me, while I was in grad school, well, let me back up a little bit. While I was still in college, I had actually ended up developing an interest in not only public health, but environmental health and environmental justice specifically. So once I got to grad school, I knew that I wanted, of course, I was going to continue doing basic science, but I wanted it to include some aspect of, environmental health. So I was lucky enough to be able to find a lab at Vanderbilt University. And this was a reproductive toxicology lab. And I was able to do a project that basically focused on the transgenerational effects of toxicant exposure. So specifically, I looked at how a father's history of exposure to a chemical known as dioxin influenced the health outcomes of his offspring, basically. And I did find that that father's history of dioxin exposure did affect the lungs and the intestines of his children, basically. And while I was working on my PhD, like I said, I was also interested in public health. So I was trying to find a way to be able to loop in a public health education with my basic science education. So I actually ended up taking, I think it was like a 3 semesters or 2 semesters of classes so I could earn my certificate in health policy. And that kind of exposed me on how to write policy briefs and just public health in general, kind of like a nice little introduction into public health. And then on top of that, during my last semester of grad school, when I was done with classes for my actual program, I went ahead and picked up an epidemiology class while I was writing my dissertation. So I took Epi 1 at the end of grad school just to kind of, get more experience in the field that I was interested in. So Epi public health. And like I said, I participated in some internships and fellowships and other activities that greatly helped me or influenced my early career path thus far, or have influenced my career path thus far. So in undergrad, I did summer research at my home institution. Of course, that was basic science. That was actually how I was able to find out that I was interested in environmental health, so environmental exposure. So during that time, we had I think it was 3 types of seeds, like basil, spinach, and some other seed. And we grew them in different concentrations of a chemical called bisphenol A and bisphenol S, BPA and BPS. And just basically, that was my first time seeing how different chemical exposures could influence growth. And I was like, well, if it influences plant growth, I can only imagine what it's doing if people are exposed. So that experience led me to pursue another experience in public health that also, allowed me to stay in basic science. And that was the Diversity Summer Internship Program at Johns Hopkins. And that is the program that introduced me to environmental injustice and epidemiology and like, you know, public health is a major field basically. So after that, I did another internship called Project Emotep, and that is like a partnership between Morehouse, Emory, and CDC. And with that, I basically got a crash course of epidemiology, public health, like intro to public health, I think intro to statistics course. And then I was stationed or like placed in a lab at Emory to do like basic science. And I also had like a mentor at CDC to do like some public health activities with. And then in grad school to further like expand on my interest in public health and epi, I continued to do internships. So I did a virtual internship with a program called Breast Cancer Prevention Partner. And basically that was like an advocacy policy internship. And I was like basically helping them create a document that it was basically like a summarized bibliography of papers that suggested that certain or different chemicals in personal care products were associated with like poor health outcomes in effort to advocate for like clean beauty products, essentially. My most recent fellowship was with Mount Sinai. So I participated as a global health fellow, and I was able to actually travel to Brazil to do a epidemiology project. And there I was looking at a cohort of mothers and infants and basically seeing looking at their heavy metal exposure profile. So looking at like how much arsenic, cadmium, mercury were in like a mother's breast milk or in an infant's urine and things like that. And then other things I did outside of like research were, like was mentioned, I was an OFIS mentor and an ambassador, which I consider like advocacy and leadership experience. I also participated in the National Science Policy Network's policy brief competition. So I got like policy writing experience. I also participated in a program called Future Leaders, Advancing Leadership in Endocrinology, which was really like a leadership symposium. And then I also did a lot of mentorship programs where I was partnered with professionals and kind of they would introduce me to other people in their field or in fields that I was interested in. And an example of that program is the SHINE program, which is hosted through Merck. So after school, I had a lot of interests. So those included health policy, health disparities, applied public health, but I had a few that were like near and dear to me. So that included epidemiology, basic science, and environmental health. But I knew that once I graduated from school that I couldn't pursue all of these things at once. So I had to narrow it down, and I chose to pursue some type of post graduate training that would enhance my skill set in epidemiology and environmental health. So with that said, I ended up having to make a choice between accepting a position at the CDC or accepting a position at Harvard. So that, and I'm including that because that's another aspect of, I guess, when and how you can integrate like basic science and public health into your career. So you can always like want to do something, but sometimes things such as like location, just its lifestyle factors can influence, you know, the decisions that you make in your career as well. So that being said, I ended up choosing a position at the CDC, and both of these positions were epidemiology training in environmental health specifically, or like environmental epidemiologist. postdocs. So with that being said, once I accepted the position at CDC, I had a little like time in between me defending and actually like the start date for the position. So I worked at Emory for a couple of months, and basically that allowed me to enhance my coding skills. And while I was there, I was able to do a project that investigated how maternal stressor, specifically maternal mental health status. So like if a mom had depression, if she had anxiety or something like that, how those stressors influence the RNA expression in her placenta. So that was a pretty cool project to work on, and it was outside of my scope, but again, it helped me to, like I said, enhance my coding skills. And now currently I am stationed or positioned at the Centers for Disease Control as an epidemic intelligence service officer, and I'm also a member or an officer in the United States Public Health Service, which is a commissioned officer organization. And I'm stationed at the National Center for Environmental Health at CDC. Excuse me. So now I just want to give a little bit of an overview about the Epidemic Intelligence Service. So if people that are, well, EIS is an epidemiology training program, and it's targeted for people with advanced degrees, so medical doctors, nurses, veterinarians, PhDs, dentists, and yeah, degrees like that, basically. And if you're interested in applying, then it's just good to know that, let's say, if I wanted to be an EIS officer in 2025, then I need to submit my application by March 2024. So basically, the application and selection process is about a year long. And once you're accepted into the program, basically you attend a conference where you get to meet all of the supervisors who have open positions, and you get to like chat with them, you know, see who you connect with, who you would want to work with. And after that, you are able to interview with up to, I believe it was 10 positions when I did it last year. And probably like a week or two after that, you receive a notification about the position that you actually matched to. And this is just from my experience as an EIS officer, but I would say that a good candidate for this fellowship would be a person with an advanced degree who does or does not have formal training in EPI. So in my cohort, there are people that have PhDs in EPI. There are medical doctors who don't have much research experience. There's a couple of people like me who have a research background, but not a epidemiology like research background. So it's open to anyone, you know, who's interested in the training. And other good candidates will be people who are just interested in understanding how government works or how to conduct research in a government setting, because it is only a 2-year fellowship. So you get a nice exposure. And then, you know, after the 2 years, you can decide if it's something that you want to continue. I also believe that people with, you know, like I kind of mentioned before, virtually any like health or research background who are interested in EPI would be good candidates, and people who are interested in improving their data analysis and writing skills. I do a lot of coding and writing in this position. And some of the things that EIS officers do is participate in outbreak and field investigations. We also write concise reports such as MMWRs, which is like CDC's journal. We give presentations to expert audiences and lay audiences, attend conferences, write manuscripts, complete different analytic projects, complete surveillance projects, surveillance evaluations, and you also get to experience interacting with external partners from state, local, tribal, and territorial regions, and much more, just, like depending on where you're positioned at or matched to. And EIS officers work at the CDC and certain public health departments. So these are just a couple of examples. So like New York Department of Public Health, Agency for Toxic Substance Disease Registry, National Center for Immunizations and Respiratory Diseases. So there are a lot of different breadths of like public health and different types of research being done at the CDC and by EIS officers. And in closing, just to kind of talk about my, I guess, future plans. So just continue enhancing my data analysis skills while I'm in this fellowship, and then apply for a full-time position with CDC. And since I'm in the United States Public Health Service, basically I'm qualified to retire after 20 years of service. So I plan to complete like 10 or more years of service at CDC, and then possibly transition to another government organization such as the National Institute of Environmental Health Science or Environmental Protection Agency, something where I can still do environmental health related work, and then, retire from the public health service. And then I'll still be fairly young, so we'll see if I might start a whole new career after that. We'll see. And just a summary of, I guess, things that I've realized along my path is that blending social justice and science into your career is not going to be a linear path like all the time. And people might tell you that it's not possible, and sometimes you may feel that way, but you just have to keep pushing through, because at the end of the day, it is possible. And it's always important to continue networking and participating in everything that interests you, because you never know how one activity can like change the direction of the year. You meet somebody, you gain a new interest, you never know. And just always look for ways to include advocacy and social justice in your research and work, even if it's not the norm, at your job or at your school. And thank you.
Wow. Thank you so much for sharing that with us. Thank you both for giving us some insight into your career paths. It's been fascinating getting to see how you respond to infectious diseases in the United States and globally. Before we move on, we just like to quickly ask that people turn on their cameras so we know that you're engaged with us. Thank you. And then we ask some of our teams that if they have any questions in advance, that they should send it to us, but everyone is welcome and encouraged to send their own questions in the chat. And then we'll call on you to unmute yourself soon. And also don't forget to fill out the attendance form, which Hassan has posted in the chat. Does everyone see it? Yeah, cool. Okay, now we'll move on to the Q&A section of our program. So we have a number of questions that were sent in advance, and a few of you from the chat will ask you to unmute yourself and ask your own questions. So we'll start with the questions that came in before tonight. So the first question was from a mentee, Ashley, and this is for both of our participants. How can one protect their health against something that is out of their control within their environment? For example, for people who live near highways, they produce many harmful air pollutants, and according to the American Lung Association, long-term exposure to traffic-related air pollution is associated with asthma in children and adults, and lower respiratory infections in children and premature death. For many families, moving away isn't always an option, so knowing how to protect your family is important. So, did you get that question? I can repeat the question really quickly again.
No, I got the question. That was like, I think someone emailed it. That was actually a really good question. I actually struggled with that one a bit. I don't have a good answer. The answer I do have though, I think that's kind of where the idea of like the equity and us all buying into this together comes into play. Because like in the example you gave, if that family doesn't have the capacity to move for whatever reason, that's like why it's on all of us to make sure that people aren't put in that position, if that makes sense. And so I'm not exactly sure what does that family do, but I think that's not a family problem, an individual problem, that's a societal problem. And so like that needs like a societal solution, like if that makes sense.
Yeah, that was a very good question. And I feel like that was a question that I asked myself a lot during my PhD, and it stressed me out. But I would say, so you cannot always control where you live. Like the question said, you might live next to a highway. You might not be able to afford to move to a neighborhood that is less polluted than that one, you know what I mean? So what you can do is try to take like other steps in your life or change other things in your lifestyle to reduce your exposure. So let's say I know that I live in a neighborhood that's polluted. Well, what do I have control over? Something I have control over is the types of products that I put on my skin. I can make sure that I try to use products that don't like contain any like parabens or other chemicals. Like I can start doing research on that type of stuff. I can, let's say, I'm trying to think of something. Let's say I have like a habit of trash burning, but I live in a polluted community. That's something I can like try to stop my own habit of trash burning to at least reduce the amount of pollution that's in like my space or my vicinity. So I would just say like taking steps like that, or even let's say you are afraid that like your soil or something might be polluted like in your yard. So taking the step to, if you want to like plant a garden bed, to invest in like having a raised garden bed where you're using, you know, outside soil versus the soil that's in your yard and stuff like that. So I would say like taking steps like that could kind of help. Well, it would help overall when there's things out of your control that you, there's things that you can't avoid.
Thank you for those answers. For the next question, I think Asha, she had a question. I think she's on the Zoom. If she's not here, I can just read her question. It was, many other people in your school most likely had similar goals and aspirations regarding public health. I see that you went to a pretty prestigious institution such as Duke, and that's her dream school. So she's wondering how you were able to persevere despite endless competition, and how you were able to garner support without extensive connections from other people.
Yeah, I mean, that is another tough question. It was definitely like a competitive place. But I guess just like you just have to remember I mean, obviously like you're competing, I guess, in terms of grades and whatever, but you have to just remember like your main competition is yourself, and you shouldn't even see yourself as a competition. Like you should be motivated by whatever is driving you to be there in the first place. And so that was kind of my mindset in being in like more hyper competitive places. it's hard to do because you're in college and you're young, but you have to identify a sense of yourself and like what you determine to be important and let that dictate, I guess, how you feel about your environment. And also too, it's not like there's like enough opportunity for everyone in a way. It wasn't like there was space for everyone who had aspirations in public health to do what they want, and that's true if like I Duke or any other school. So to be honest, it is like a competitive place, but you just have to be in the mindset of just like focusing on what matters to you in order to succeed at places like that.
Very good advice. Thank you. Our next question is from Therese. I think she's in the Zoom, so she'll ask her question.
Hi. So my question was, have you like been interested in like the program like of Doctors Without Borders? Because I feel like it's really an interesting method for like Western medicine to be brought to more like disadvantaged societies.
Yeah, so not that program in particular because it's for medical doctors mostly. And I'm more interested in basic science research. So when you like kind of think of science in terms of like beakers and a white coat in a lab, that's more what I do. But that same spirit applies, like especially with infectious disease research, like I mean, as we talked about, because of just the importance, like globally, like we do spend a lot of work in terms of like you can so like I know a lot of malaria labs, for example, they'll go to sub-Saharan Africa and collect samples, and they engage in like capacity building in those places. So that's like a huge problem, is like we're here at Harvard and we're going to these like developing countries and we're doing research on like the pathogens in their regions, but like it's important to also build up their own capacity to do research there, if that kind of makes sense. So that's kind of how that looks like for a basic science aspect. So like next week, actually, I'm going to Cape Town, South Africa for a strep pneumo conference. And a part of that conference involves like going to the university in Cape Town and learning their lab methods, and we teach them our lab methods. So there's like a back and forth between like because we don't want like kind of like that ivory tower like savior complex to take place either. Like these people who are burdened with these diseases, they also have input and like value add in terms of like how we can approach combating them.
Thank you for your answer. We only have time for 2 more questions, so we'll speed run through these. This is for Dr. Rump. Sanea, I think you had a question in the chat. Or a question you sent earlier.
The question I sent earlier was for Dr. Stanley. I noticed in your presentation that you had one graph that had like a bunch of dots and like a middle line. I saw that in a few of my classes, and I was wondering if you could explain how to interpret that in a general sense, because I know that you briefly explained it in your presentation in the context of what you were talking about, but I just wanted to know how I could interpret it as a like receiver of that.
Yeah, no, you're absolutely right. I did not explain that at all. When I saw your question, I was like, I should have done that. But each little dot would have been like an individual strain of bacteria. Like I said, so we assessed like hundreds of strains from Vietnam. So each dot would indicate an individual strain, and they were grouped based on like their genetic profiles into 3 different groups. And so that middle line you're referring to is just like the median like fitness in the presence of an antibiotic. So if it was like down here, it means they're super sensitive, like they're being killed off. If there's not that many representatives of them, but if it's like super high up, they were more yeah, thank you. Yeah, so these are like here, these strains are super fit in the presence of this antibiotic, and these are super sensitive. And this was just an assay to screen the phenotypes for a lot of different strains at once. And we could see these patterns, like these strains with this genotype, this L1 genotype, were very sensitive to these antibiotics. And so it suggests that maybe we should use these antibiotics to treat people in Vietnam, because these strains are enriched in Vietnam in particular. Hopefully that helps.
Yeah, that does. Thank you.
And then we have one last question from Ishan. Am I pronouncing that right?
Yeah, so this one is for Dr. Stanley. In your presentation, you talked about how you would tailor treatments to different regions based on the genetic profiles of the disease vectors. How do you plan on addressing the problem of cost? Because if you tailor the disease treatments, then they'll probably cost more, leading to less people being able to afford or access it.
Yeah, that's a great question. So actually with the way tuberculosis is treated, it's like a cocktail of a lot of different drugs. And the drugs I showed are drugs that already exist on the market. So like we don't have to create new drugs, we just have to optimize which particular drugs are given to which groups of people, if that makes sense. So there's actually no additional cost. And that's what makes this like so frustrating, is that we could make treatment a lot better for people with very minimal effort. Like we don't have to discover new drugs. And that's where like that injustice comes in. Like there's no motivation to do this because we don't really care about the people who are dying of tuberculosis. If this was a problem in the United States, we would have been done this. But like we don't actually have to make new drugs. We can just, these are drugs that already exist on the market. We could just take the extra step of matching the drug to the patient. And that's what we already do with cancer. This is not like a new paradigm. It's a new paradigm in terms of infectious diseases, but the paradigm of like sequencing and then matching your drug to fit that's already how we treat cancer. We just want to apply that same approach to infectious diseases. But you're absolutely right. And a lot of TB drugs are donated by the companies that make them because they know that the people who need them can't afford them. So that's also at play as well. But that's a great question.
Okay, thank you. Well, that's all the time we have for tonight. Thank you for an incredible talk. We are grateful for Dr. Stanley and Dr. Rump for spending this evening with us. To our audience, you can extend your appreciation in the chat with some thank yous. And just some quick announcements. Mentees and mentors, don't log off. We'll have a few minutes devoted for you to ask us any questions, and please complete the attendance form as well. That's in the chat. We can send it again if it's lost. And thank you for attending tonight's community talk. Please visit ourfuturescience.org. Follow us on social media to learn more about our initiative, future events, and educational campaigns. I posted the links in the chat. And if you can't stay on, we just want to send you a few quick reminders for upcoming deadlines for mentees. Module 5 is due this Sunday, March 10th. This is where you can pitch your final output projects and solutions for OFIS approval. One member from each of your capstone project can submit their responses in Module 5 in Canvas. Check the assignments tab for submission details. And then for mentors, we're excited to see all your lab videos, which are due March 15th. And we really appreciate you sending it in early if you can. So those are all the announcements for tonight. If you have any questions, stay on and have a great night, guys.
And we'll share Dr. Rump's lab tour from when she was a mentor too. So you'll get an extra one.